Western blots are a powerful analytical tool used by investigators worldwide in research and development, pharma, and food industries. Western blotting technology allows you to separate and identify individual proteins from a complex mixture of proteins that were extracted from biological samples. Depending on what you want to accomplish with your study, you can generally choose from one of three types of Western blots: qualitative, semi-quantitative, or quantitative. Now let's look at each of these different types of Western blots. A qualitative Western blot is a simple visual assessment of target proteins. An example of a qualitative Western blot would be if you're studying a novel protein and want to see if that protein is expressed in the cells of interest before performing a more full-scale quantitative Western blot. As shown in the example blot below, the positive lane shows the target protein band. The negative lane, however, does not have any visually identifiable band. This type of quick visual assessment may be enough for you to proceed to the next steps in protein quantification. Simply eyeballing for differences in protein expression would be classified as qualitative Western blot. In this example blot below, we can see that one lane has a target band with faint signal compared to the next lane with much higher signal. This too is a quick visual check to look at the effect of a drug on target protein expression. To review, the qualitative Western blot may have a place in your studies for a quick check for the presence or absence of the target protein, as when screening a patient sample for Lyme disease. In this case, you would evaluate the Western blot to have the right number of bands at the right place using a simple plus or minus test. The blot is simple and straightforward, since there is no quantification required of those bands. Qualitative Western blots can also be used to look for changes in protein expression upon a drug treatment. For example, you may want to determine if the concentration of the apoptosis-inducing drug in your study affects expression of your target. Again, you're not quantifying the exact change, but simply visualizing it. A qualitative Western blot can even be used for studying DNA-protein interactions via Southwestern blotting, where the exact concentration of the protein of interest is not crucial.